was on the bus today and I was talking to this lady that went to she said she's studying biochemistry. So I asked her a question. Is there like some type of chemistry or like chemical going on when cells are splitting, stuff like that, right? And so she couldn't remember it. I just watched the video, so kind of got me going. So so I was watching a video, right? And so it had to do with amino acids, right? So basically what happens. So there's three different kinds of amino acids, or three structures or something like that. I think it's like monopeptide, monotide, polypeptides, and then you got multipeptides or something like that, right? And so based on their structures and stuff like that, right? So what happens is they usually consist of hydrogen, it's hydrogen, nitrogen, and carbon, right? And so I think the hydrogen, something like that. Well, I don't know, right? I can't remember fully, but I just know when amino acids. Well, in order to get amino acid, you got hydrogen and nitrogen. You got that bond, right? So that's the amino, and then so the acid part comes from a different same structure, but instead of the hydrogen and the nitrogen connecting, you got the oxygen or something like that. You got oxygen connecting to the hydrogen. So then now you got it releases H2O. Something I just remember something about that. It releases H2O, right? Water. So there's like four different stages, right? You got the primary, you got the secondary. So this has to do with amino acids, right? So you got the primary, which is just like the DNA itself. You got the secondary, which is like the backbone of the amino acids. You got the tertiary thing, which is like basically I think it's the side, right? The side of the amino acids, and then you got the fourth one was like oh, I forgot what the fourth one was, but so then you got four stages of that, and so I was wondering if DNA, right, the thing that's inside of the DNA, if that is just made up of proteins, basically, right? Essentially, a bunch of amino acids, right? So that's what it is. It's a bunch of amino acids that are put together, right, a certain way, because like I guess there's like 7 different ways that amino acids can either bind or not bind with each other. So like if you got a hydrogen on the side, you have a hydrogen on the side, and then you have an oxygen on the other side, then you got a bond right there on the side, right? You got a side bond, which is which is the called tertiary, right? During the tertiary, if you have a hydrogen and an oxygen next to that, then that thing's going to want to fold inwards because um, they're attracted to each other. You got a positive and a negative. However, like if they're like hydrophilic too, those are going to be together because I think that's the reason why the hydrogen and oxygen right there. But then you got things that are maybe like that don't like water. They're called like hydrophobic, right? Um, this is what the things were saying. So what I'm wondering um, is that the reason why telomeres are shortened, right, is because for some reason um, during during um, during the process of the amino acids coming together to make up that code, right? Basically that amino acid chain or that protein, somehow, some way, um, one of them doesn't fold right, right? So you got like maybe instead of like a hydrogen in one spot, you got an oxygen in there instead, or you got like, and so what happens? It kind of just messes up that protein, right? So then, when that cell is finally formed, it can't. It knows how to like live in that area, right? But then, but it doesn't know how to survive, right? So 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 what I'm thinking is that um, that the reason why people are like people are basically deteriorating. They basically why people are dying or getting these defects and stuff like that. Um, is because of epigenetics, right? Um, so epigenetics, they're still doing a study and stuff like that. It's still like new research, but if it's true, right? Which I'm assuming it is true, right? So what happens is like the environment kind of like causes the DNA, right, to turn on and turn off. And so what I'm thinking is that due to environmental changes, um, these proteins, right, these amino acids that make up the DNA, because that's what the DNA is a bunch of amino acids um, that are bind in a certain way to make up a primary code. Basically, it's like a code, right? Your DNA is basically like a code, and so what happens is like, um, let's just say like for 5 years, you you live I don't know, somewhere where it's cold, right? And so what happens is, and then you move, right? You move, and then it's hot. So what happens is that these proteins, say like if you're born, basically like if you're born, right? If you're born in a cold region, and then you move to a hot region, these proteins, these DNA, right? These DNA strands, basically like amino acids, are coded for cold weather, right? So they're bending in certain ways. They're folding with each other, against each other. They're just basically like structuring themselves in order to live in that environment, right? So then when you move to a different environment, the protein, basically that cell, right? That DNA in that cell, which is amino acids, they're just like, well, wait a minute, like I I don't I don't need to fold this way. I don't need this. I don't need right. So like, let's just say if in the cold, let's just say if it made a U shape, right? Let's just say the whole, like for easy sake, it could be way different, right? But let's just say, like, make an easy observation and just easy explanation, right? So let's just say during the cold area, it's bending like a U or a V, however it looks like, right? Like a U, right? And so then that's what they're bending that way because like certain molecules or whatever, right? The amino acids, the molecules, they like each other or whatever, right? But then when you get to the hot thing, it's like, well, the DNA, that cell is just like, well, the DNA in that cell is saying like, well, dude, I don't need to fold that way because um, because of the environment, right? So then it folds like a C, right? So then now you got that shift, you got that different change, and so what happens in order to make that change? Let's just say the DNA, right? When it produces itself, when it starts splitting um, into mRNA, then it's saying like, well, hey, there's certain amino acids, right, that don't need to be together, right? So then it folds a different way, give you the C shape, and I think that's how it causes mutations, right? That could be the way the epigenetics are working, and so like if you're I think so. If we're constantly switching environments or environments are constantly changing, then then um, that's why we get a bunch of mutations like cancer, sickle cells, um, bacteria, right, viruses, um, because it's the change in environment that is ultimately ruining us. So if you think about like a fish tank, right? Um, or just like a habitat, right? Like I don't know. The best way I can put it is like a fish tank. Okay, so that fish tank has to keep a certain temperature, right, for certain fishes, right? Because there's certain fishes that are like cold, some are tropic, some are freaking like salt water, right? There's different types of fishes, or fresh water, and then there's salt water, and the temperature can change, right? And so fishes, right? Not fishes, but fish live in certain areas that basically allow them to thrive, right? So, for example, dude, like, um, I don't know too much about fish, right? But let's just say, like, I don't know, fish that I used to have like, they're called corridorises, right? Corridorises. Um, they like their temperature anywhere from like I don't know 60. I think it's like 68 or something, like anywhere from like 65 to like 68 or something like that, right? In that temperature. And so if the water gets cold, they start stressing out. If the water gets too hot, they start stressing out. Um, and so um, and so what happens is when they start stressing out, then you got you got a bunch of like hormones that are like trying to like they're flooding the system and blah 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 blah. And so that's basically like us, right? With this change of weather, the since our weather uh, isn't like kind of like all year round weather where it's like kind of the same, like, right? So you wake up one day, it's 60 degrees. By the time you go to sleep, it's like 40 degrees. But and then so let's just say if it's constant like that throughout like the whole year, 60-40, I think we would have um, less mutations. Our telomeres were not shortened. Uh, our like the basically the amino chains, the amino acids, right, that make up these proteins, they they would be okay. They would be copying each other just the same because there's no changes in temperature. There's no changes in anything, right? And let's just say some of these amino acids, um, right? They work different in certain areas. Like if if our whole body is composed of cells, right? And these cells are different. And some make up organs, some make up tissue, some make up nerves, right? But they're all cells. They're all cells, right? And so let's just say like our lungs, right? They they're bending and shaping a certain way because of the pollution, right? And so then when you change that pollution level, right? Let's just say it goes from high to moderate, right? Then you got like proteins trying to change what that freaking trying to adjust toward trying to adjust themselves towards that increase or decrease of pollution. Um, and this same thing with weather, right? So you got maybe the skin pigments, right? The skin that's one of our bigger organs, one of our biggest organs, or is our biggest organ. You got the skin trying to trying to defend itself against these different types of weather, right? And so then the amino acids they change, they bend different ways because of the weather. So that causes mutations, and it's I don't think it's the fact. I don't think telomeres are the telomeres, right? They caught on, right? So they know that telomeres are shortening, right? Which is basically like it's your genetic code, and every time a cell reproduces, it loses a piece of itself, right? And that's what it's a telomere part. And so, what I'm thinking, right, is basically what I just said. Is it's not losing a copy of itself. It's the amino acid, right, or that DNA, basically, right, is reconstructing itself in a way for it to survive in that environment. And so then that's why um, you're losing. That's why your telomeres are shortening because it's getting rid of things that it doesn't need in order for it to be fit for that environment. And so and since all environments are constantly changing, right? Oxygen levels, freaking weather, a bunch of stuff, pollution, it all plays a factor in the way our genes express themselves. And so when these things are unstable, then that's and so since these things are unstable, they're constantly changing. Like pollen, Jupiter, um, freaking what is it? Freaking what's the other one? It's like air quality. Um, since they're constantly changing, it's messing up our biology. It's constantly messing up our biology. It's constantly having, maybe not constantly, but it's having. Let's just say if I don't know. Let's just say if you have 10 cells, right? I know there's probably more, right? But let's just say if there's 10 cells that make up the liver. Let's just say that 10 cells. I know there's probably more, or whatever it is, right? I know I'm not saying this right now, but let's just say if there's 10 cells, and then so um, so your liver gets a toxin, right? And it's like, oh shoot, like wait a minute, like I never experienced this before. Like in order for me to survive this, I need to get rid of this protein, um, right? This molecule, right? Basically, this amino acid or the amino acids or whatever. It's basically like hydrogen, nitrogen, right? It gets rid of a nitrogen or a hydrogen or a carbon, right? And so in order for it to fold a different way and be able to like um, to digest or basically like consume whatever it is, right? So so what is happening is like I said. So then when it changes, right? And let's just say in order for it to express itself that way, but not just express, not to become a phenotype, but like a genotype, right? It's like a non-observable. Um, then it's like, hey, well, I need to get rid of a carbon here, and if I get rid of a carbon here, um, then I'm not going to be able to bind with, I don't know, with something else, or just our like pulse or not bind, but maybe just like maybe repel, right? Because if they're folding, they're like unfolding them. If they're folding close to each other, then sometimes they go away from each other, right? So they either basically like contract, like fold towards each other, or move away from each other, right? And so if it's like, hey, I need to move away from this person, but in order for me to do that, I need to get rid of like a carbon, or I need to get rid of a hydrogen or a nitrogen, right? So then that change, so then that's the change in the telomere. That's what's changing. That's what's changing the genetic sequence. That's changing the genetic sequence. So then now that cell has a different code. It has a different code. And I wonder if that's true. I wonder if, because the professor from Bio 6 is saying that DNA doesn't change, it just varies. It turns off or it turns on. But I don't, maybe she's right, but I think it can't be, because well, maybe the original one stays the same, right? The original, like say when it's going through mitosis, right? Whatever, if it's going through like the mitosis, basically, right? When it's going to go through mitosis, the interphase and all that stuff. Then the original one probably stays the same, right? But then the new one becomes, it's not an exact duplicate, because for the simple fact that the protein structure has changed due to the fact of it wanting to survive. It wants to survive, so it's going to create a suitable amino acid, right? So basically, like, how would I say it, dude? It's going to create a suitable like replication, but it's not going to be exact, right? And so then that cell has a different like a sim, like maybe it's like a, I don't know, it's like if you were to put it in a percentage, maybe it has like a 0.00000000001 variation, right? It could be longer, right? The variation could be very minimal, to the point where it's like it's noticeable, but at the same time it's like, oh, like you wouldn't even be able to tell. It's like kind of like a needle in the haystack type of thing. Like, hey, we know that there's a needle here somewhere, but we just don't know. It's so like hidden that it doesn't really mean anything, right? It's kind of like I don't know, like if you get like one little piece of grain of sugar, one little small grain, I'm talking about just one. If you're able to grab one grain of sugar and throw that in your water, do you think you're gonna be able to taste the difference? You're gonna be like, oh, I can taste that single thing of sugar, right? A single grain of sugar. No, you're not gonna be able to know just until you put like, I don't know, but like 10, maybe 20, maybe more, right? Then you start noticing. And so that's what happens with the mutations, right? So then you got a mutation. You got a gene that mutated. Not a gene, but a cell that mutated. But the thing is there, it's almost the same, but we think it's not. We think it's like, oh, well, it's just a little small change. It's not gonna hurt us. But then those changes build up over time. Then now you got a problem, right? Because now it's like, well, wait a minute, like, like, now I noticed the difference. Now I noticed, like, I went from, like, basically, like, let's just say if it was bending like a U, right? It was a U, right? And then and so maybe I was doing this. The U shape is the original, and then it's like it tilts a little bit, right? And then it tilts. It keeps tilting with each different. They're replicating itself, but it's just changing slightly, right? And it's over time, it's changing, it's changing, it's changing, it's changing, it's changing, it's changing. You're like, and then it gets like to right here, and then you're like, well, wait a minute, holy crap, it's mutating. It's not complicating itself, whatever, right? And then over time, it keeps changing, changing. And you're like, holy crap, look at that! It went like a whole 90 degrees, right? It's like 90 degrees. And so then that's when the change becomes significant because the change is noticeable, and that's when you probably get like a lot of mutations and all this stuff. And so and so that's the reason why you can't reverse. You can't reverse aging. You can't reverse um, any type of things that are um, that are damaged, anything like that, because its original format has changed so much that origin, right, that origin of that cell is no longer original. It duplicated itself so much that 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 it's no longer normal. We may think that it's normal. Oh well, yeah, that's how it's supposed to be, right? That's how it's supposed to be. But in order in order to go back in order to go back in time, right? So not time, but just aging, right? To reverse aging, you would have to get someone's um, DNA sample. Every single you would have like if there's over 3 billion cells or something like that, right? If there's over 3 billion cells in the body, or maybe just in the neural network somewhere, I don't know. But there's like billions of cells, and you would have to write down every single thing about it. Like, cell one has, I don't know, like, how many how many amino acids? I don't know. Like, you would just say like it has 2 amino acids. Then cell two has 3 amino acids, and cell four has blah 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 blah. You would need to know how each amino acid is connected, right? So if it's a mono amino acid, if it's a polypeptide amino acid, if it's a multi-peptide, right? And you would have to look at every single bonding. You would have to look at all 4 stages of that bonding, right? Or basic, yeah. You would have to look at its original bonding format, which is a primary, right? Basically the code, right? It's a primary code for each one. You would have to look at the primary code during that time. So, like, if let's just say if they did that to me when I was like, I don't know, 10 years old, right? The bends in my proteins are going to be way different than they are now, from then versus now, right? And in order to restore myself to the age of 10, then I would have to get those bends back in place. But in order to do that, you would have to know where those bends are at, right? You would have to know if some of the proteins, right? Like, I don't know, like you have if it's like a carbon, if it's like a hydrogen, carbon, carbon, right? If it's like that hydrogen, carbon, carbon, or if it's a nitrogen, carbon, hydrogen, right? If you got different sequences like that, then like it's like, okay, well then I got to restore myself back to a hydrogen, carbon, nitrogen, and a nitrogen, carbon, hydrogen, right? In order to get that fold back, thus causing your gene to get back to its original self, that cell to get back to its original function. That's the key. That's how you reverse aging. That's how you cure someone. That's how you you find medicine for people. And then that could go the same thing for like these neurons. reason why we think that we can't um, restore brain neurons, right, brain cells, is because we don't understand the amino acids, right? Like, dude, like, what happens if like are these neurons? They're just like constantly changing, for the better, right? So it's like, oh, I learned something new, so it folds. It folds. It's like, hey, well, I don't need this. I don't need this. This like, I guess this. I don't know. You would call it a molecule, right? I don't know if that's it, right? A molecule. Well, I don't need this carbon molecule, or I don't need this hydrogen molecule, or I don't need this nitrogen molecule. Um, and so then it loses it, and so then it makes that connection better, right? But then, like, if you start like polluting yourself with different stuff like microplastics or like drugs or anything, right? Then these things start folding a different way, right? And it's just like, oh well, I folded so many different ways that I can't even make a connection, right? It's that connection is going to be lost. And since we don't know the original, and I think that's right. So I just thought of something. So that's why it's like that change happens way faster than 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 maybe the proteins inside of our neurons change way faster than the proteins in our liver, than the proteins in our skin, right? Like basically the DNA, right? So if the if if the neuron carries a DNA inside of it, because it has a cell body and it has DNA inside of it, then that DNA inside the neuron is changing so fast that by the time we catch that like that that like abnormality, right? Or just basically like if we like, oh well, this person's like uh, has a disorder. Basically, when we catch that disorder, it's because it's too late. That neuron, it could be multiple neurons. It could be a single neuron. It could be multiple neurons, right? That change so much that 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 we can't reverse it because we don't know its original state. We don't know its original state, and if everybody is kind of different, right? We're kind of wired different. We think different. Then let's call it. Then you wouldn't know, but maybe we could copy. But then, if you copy someone else's neuron, basically the DNA. If you copy someone else's like DNA, like if we pinpoint like the neuron, let's just say if I'm having schizophrenic episodes, right? If I'm schizophrenic, right, and I'm having episodes of hallucination and uh, and and I'm delusional, right? So I'm thinking. So what happens is like hallucination. Well, what is that? That has to do with the occipital lobe, right? It has to do with your occipital lobe, and then from there, right? Then it goes to then from your occipital lobe, it goes to maybe to your hippocampus, the wiring, right? Let's just say like it goes to your hippocampus, and then from your hippocampus, it goes to your prefrontal cortex, where it's like where it does the processing, right? It's like, oh well, did I see something? And then like somewhere along those lines, um, it can skip, right? It could be like, well, maybe from the journey from the occipital to the hippocampus, it gets changed. That information gets changed. Like, oh, I seen something, right? And so the hippocampus is like, oh yeah, I remember it, right? But in order for me to remember it, let me go back to the processing place where I remember where, right? Let me go back to where I learned it at, and that's like the prefrontal cortex. And the prefrontal cortex, like, yeah, that's that. And it goes back to the hippocampus. The hippocampus is like, oh yeah, I remember now. It's like it's a rabbit or whatever, right? And so then that information goes back to the occipital lobe, and you're seeing a rabbit, right? So that could be the form of hallucination, right? Um, and so then delusional, right? Are you hearing things, right? You're hearing things. So then that sound, right, goes to the temporal lobe, and then that sound is passed on to the hippocampus, and so that sound could be changed, right? It's like, oh, like it gets changed. It's like, oh, I didn't hear a scream. I didn't hear a scream. I heard a voice, right? And so um and so then it goes like, well, where did I hear that voice? And then it goes to the prefrontal cortex where it's processing and all this, maybe where it processes it, maybe it's like to other regions and stuff like that. Then it goes back to hippocampus and it was like, oh yeah, it was Michael's voice. And so then it goes back, and then it goes back to hippocampus, like, oh yeah, and it's like, oh yeah, well, like, it's Michael's voice, right? And then, so then that pitch change, right? So that could be the explanation of schizophrenia, but it can not just be one neuron, right? A bunch of neurons could be a factor, right? And maybe some neurons, what happens is that that it could be the fact that the neuron wired with another similar neuron, right? So what happens is, like, the terminal button deattaches itself from its original to the synapse, right? So let's just say these are the primary, right? So this is how it is. The first, you got the terminal, you got the synapse, and then you got the receptor. But then for somehow my thumb switches with my middle finger, right? So it's like, hey, like, I'm gonna establish a connection with with a different receptor. And so what happens is, since they're so similar, right? And then so what happens is, like, it's slowly, gradually. So it goes like this, and so this one starts moving away, and this one starts moving closer, and so now you got you got two neurons interacting with each other, which forms a new connection, and that new connection could be the misinterpretation between the voice, or the misinterpretation of the image, right? It's like it could be something like that. But ultimately, what I'm trying to say is that um, these amino acids, those are the molecules, carbon, hydrogen, nitrogen, oxygen, are basically the ones that are controlling. Those are them. That's what makes up. I don't know precisely if that's what it is, but it's from what I learned, if that's what DNA is, it's just a bunch of hydrogen, nitrogen, and carbon along with oxygen. Then, in order to reverse aging, or not to age at all, you need to get a copy of every single cell inside you. You need to know its original code in order to stay like that, not to age at all. If you can somehow reproduce that same protein every single time without losing it during the mRNA stage, then you wouldn't have mutations, you wouldn't have aging, you wouldn't have the loss of anything. But that's a lot of work. They said there's 20 to like 50,000 amino acids, and so you would have to know every single one, 20 to 50,000 amino acids structures. I think that's what it was, and times that by 3 billion cells, right? 3 billion cells, that's a lot of different changes. A lot. I mean, that's a lot of storage that you would have to freaking do. 3 billion times, let's just say for simple, easy mathematics terms, it's 30. You got 30 million amino, 30,000 amino acids times 3 billion. I don't know, maybe what is that, like 150, like would it be 15? Would it be 15 billion? Maybe more. I don't even know. I just know if you cut off the zeros, it would be like, it would be 15 because 3 times 5, and then if you add all the zeros back together, right, it would probably be over a billion, over 15 billion. I would say that, right? It would probably be over 15 billion, maybe like, I don't know, 15 trillion, 15 billion, something. I know the numbers will be big, and to be able to write down all that code would take forever. Take forever. You would need something that can work day and night, writing down those codes. And even at that, like if you're writing down a certain amount, let's just say you could write down half of your code cells, right? The next day, let's just say if you could write like half of the cells DNA sequence, right? You could write down half of that, but then by the next day, those things are going to be changed. Those things have changed because the different types of air qualities and mold, these different factors that may play a role in the change. Right? And not only that, just different feelings and emotions and all these different things play a factor too. Right? It's just constant change, and I think that's why we're deteriorating. Because it's just the changes of everything. Changes of everything. From what we breathe in, what we think, the stress, the excitement, a bunch of different things. The bonding, right? These all can play a factor in the shifting of these